Um, Great. So thank you for the introduction. I would like to start off by thanking the organizers for this beautiful symposium and for letting me present my research. I'm also very happy that I'm the only SARS-CoV-2 presentation, meaning that the world is going back to normal slowly but surely. So I'd like to start off by saying that interferon is a cytokine secreted from cells upon infection, and at least in vitro, it does a very good job in restricting the infection of SARS-CoV-2, which is the agent that causes COVID-19. And in that image, you can compare mock-treated versus interferon-treated cells upon infection with SARS-CoV-2. You can see that upon treatment with interferon, the cells become completely resistant to viral infection. So interferon induces a very strong non-specific antiviral state in affected cells. And it does so by inducing the expression and translation of a large group of genes, collectively termed as interferon-stimulated genes, or in short, ISGs. And those ISGs are important because they sit in the core of the antiviral response of cells to viruses. And their function was shaped by coevolution processes with their cognate viruses. So when we uncover something interesting about the biology of ISGs, we learn something about when and where viruses are most vulnerable. And of course, this kind of knowledge can lead to multiple new avenues for therapeutic approaches to viral infection. So how we should study those ISGs? The study of ISGs have several challenges that we want to address. The effect of ISGs upon expression for some ISGs can be deleterious to cell viability because some ISGs are antiproliferative and pro-apoptotic. There are also very much cell-specific antiviral effects. So an antiviral gene in cell type A will not necessarily be antiviral in cell type B. Also, when we infect cells with viruses, those cells usually secrete interferon, which will create a confounding effect on our readout, which is specifically true when you try to screen for the antiviral effects of ISGs. So we wanted to address those challenges using an expression system capable of high throughput screening of ISGs with antiviral functions. Our expression system is based on a CRISPR activation technology called the SANTAG technology. This CRISPR-A system has two components. The first one is a catalytically inactive Cas9 protein fused to an epitope scaffold. The catalytically inactive Cas9 is a guide directed to the transcription start site. And the SANTAG scaffold allows for the second component of the system, which is an intracellular single-chain AV antibody fused to a transcriptional activation domain to be recruited to the transcriptional start site. So upon binding of the Cas9 protein to the promoter, multiple transcription activation domain will be recruited and create a very strong transcriptional response. We adapted that system to allow for inducible expression of the intracellular single-chain antibody, so we can precisely control the timing of the expression of our target genes. So to make a very long story short, I will show you how this system works. We deployed that system in A549 cells because we wanted to have something which have a physiological relevance to respiratory virus infection. But we did more than that. We also deployed that system in A549 cells that are knockout for their STAT1 protein expression. The latter cell line is an isogenic cell line that can secrete interferon but not respond to interferon. In that experiment, I'm showing you, infection of VSV expressing GFP marker protein as a marker for productive infection in our system. These are flow plots, so the x-axis is the percent or is the intensity of GFP. You can compare the blue histogram, which are cultures that express guides targeting the activation of MX1, a very prominent antiviral genes, to cultures that express a non-targeting guide. You can see that without any kind of viral infection, regardless of treatment of the cells with DOX that induces gene expression, there is no apparent increase in GFP. When the cells are infected with VSV GFP, we can see a very strong accumulation of GFP expressing cells, although it's very different from the wild type and the STAT1 knockout cells, which can teach us something on what is the relevance contribution of endogenous interferon in that system. When we add doxycycline and induce the expression of MX1 or a non-targeting guide, we can see that the wild type cells were completely blocked viral infection, as you can compare between the blue and the gray histogram of the top plot. The infection in the STAT1 knockout looks looks somewhat different. While most cells are infected, we can see a dramatic effect on the intensity of the GFP marker. The STAT1 knockout background gives us a very rare opportunity to actually look on what is the antiviral capacities of MX1 outside the full-blown interferon effect upon viral infection. So this is how our system worked. It is an optimized Santa CRISPR activation system for studying antiviral genes. We engineered it in a clonally selected A549 lung adenocarcinoma cell lines because we wanted to work with respiratory viruses. It is capable of robust and specific doxycycline inducible gene expression, so we can control when ISGs are expressed. It is also deployed in STAT1 knockout background, so we can measure the sole activity of ISGs. Finally, it is capable of robust gene activation that can restrict viral infection. So I was very lucky to generate this system around February 2020, just before COVID hit. So when COVID started, we thought about employing our resources here at the Icahn School of Medicine to conduct a focused CRISPR activation ISG pooled screen for SARS-CoV restriction factor. The outline of the screen is as follows. We express a library of ISGs in our SANTAG cells that now express also ACE2 to facilitate SARS-CoV-2 productive infection. Our library contains 414 ISGs, 3 guides per gene. And because we want to search for viral effectors, we excluded all transcription factor, interferon genes, and pathogen recognizing receptors. This is a cytopathic effect-based screen, so we infect the cells with a high MOI of SARS-CoV-2, expecting that protective guide RNAs will be enriched in the survival population. So after viral-mediated cytopathic effect, we extract the genomic DNA from the surviving cells and prepare genomic RNA libraries, followed by analysis of relative enrichment or depletion of guides in the surviving population. We use an algorithm called MAGIC, and this algorithm allows us to use a more elaborated design matrix that takes into effect the effect of the virus, the effect of the docs, as well as the effect of the different single-cell clone and the different biological repeats that we did in the experiment. So how the results look like? We will get from each cell line, the wild type or the STAT1 knockout, a 2-dimensional graph where each dot represents a single gene either enriched or depleted in the screen. We get a set of beta score, and beta score kind of describes how the gene was selected during the screen, where a positive beta score in the y-axis allows us to know about genes that were enriched, and the negative score over genes that are depleted. And we can consider those genes to be antiviral or proviral in our experimental design. The x-axis describes genes that were selected positively or negatively only by the effect of docs. So it allows us to see what is the effect of a single ISGs without any viral infection. And we can learn something about ISGs that are anti-proliferative or pro-proliferative using the x-axis. So we can see that I just highlighted here genes that were significantly enriched or depleted in the both background. Eventually, our screen uncovered more than 30 novel ISGs with restrictive capabilities on the infection of SARS-CoV-2. And of course, we were very happy to see that a large portion, that some of the genes that were highlighted in our screens were also previously been annotated as antiviral or proviral towards SARS-CoV-2 or coronaviruses in general in other kind of experimental methodologies. Kind of validating our experimental procedure and selective pressure endured on the cells.
Oded, 1 more minute.
Yes, I will be done in a second. So, of course, after that, we had to validate those hits, and we did something very close to our screen. We expressed single guides targeting screen hits, and we did it by inducible CRISPR activation. So here, the y-axis is the percent of infection compared to dogs off, and you can see that the red dots are significant hits that we can validate in focused ISGs targeted guide. We did it by CRISPR-A, but we also did it in cDNA overexpression, just to not to overlook any kind of non-specific targets of CRISPR-A, and we could validate most of our hits. I'm just going to say it very briefly. We are following through also genes that were found to be anti-proliferative or pro-proliferative, because we have some very important highlights here of genes that we know that affect cellular proliferation and could be very, very, very relevant to different kind of cancer studies. So I've described a tailor-made CRISPR activation system capable of ISG screening by answering different challenges in studies of ISGs, and we conducted a focused CRISPR-A, identifying more than 30 candidates of ISGs that can restrict SARS-CoV-2. We validated multiple of those hits, including OS1, which is now known to be a very prominent antiviral SARS-CoV-2 factor, as well as other such as cathepsin S, and identify putative ISGs with putative functions in cell cycle regulation. Very briefly, I would like to acknowledge my lab, especially Brad Rosenberg, for his inspiring guidance throughout these troubling times, and all other lab members outside and inside BSL-3. We are also very much in the lookout for postdocs. You can scan the QR code in the bottom, get to the laboratory website, or find me on Twitter, on LinkedIn,